In our final video on our series regarding understanding mutations, we're going to talk about a rather interesting family of mutations that are what we call the frameshift mutations. Now, before we look at actual DNA, we can use this sentence here, conveniently made out of three-letter words, I wonder why that might be, as an analogy to understand how frameshift mutations work. So if we read the sentence, it quite clearly says, the man saw the dog hit the can, end. It has a beginning, it has an end, and all of the words in the sentence are completely understandable. So what we are going to do is we're going to insert one letter into the third word here so we can see that this E was not there before, so we're going to put that E right here. Now, remember the rule about how we read these sentences is that you can only read words three letters at the time, and in which case, if I attempt to read this, it would be the man see what edo git eka nend. Obviously, everything after the word see is no longer understandable unless I've unintentionally cobbled together sounds that make up a non-English language. Feel free to inform me if I have actually done so. Now, a different way that we can create similar nonsense sentences, at least in English, is if we take the original sentence and delete one letter. So in this case, we're going to take the N in the first sentence at the end of man and delete it, and if we follow the same rules about you can only have three-letter words, this sentence reads, the mass ought head og it tech ein d. Obviously, everything after the is no longer understandable. So, as we can see here, if we use this analogy where each of the three-letter words represents a codon, because codons are also exactly three bases long, we can either disrupt the meaning of a sentence through an insertion or through a deletion, which is why we collectively refer to these types of mutations in DNA as indel mutations, because what they do is disrupt the meaning of the genetic code in these DNA sequences by disrupting what we call the reading frame. So reading frames are easy to understand if we actually take a look at real English words, but what exactly is the reading frame when it comes to understanding DNA? So if we look at the DNA that occurs before the wild type or before mutations, which as Freudian slip has revealed is what we call wild type DNA, meaning before mutations have occurred, we know that we can start reading the actual protein sequence as soon as we find the first start codon, which is going to be AUG right here. So here we need to go from our DNA sequence and transcribe that in order to make mRNA, and as soon as we find our first start codon, we have identified our reading frame, because each new amino acid comes exactly three bases after the first one. So AUG is three bases. Here is the boundary between one codon and the next codon. So here is our reading frame where every three bases after the start codon corresponds with another codon. Now, in our sentences, this is why we can only read each word being three letters long regardless of what letter starts with it, which is why inserting a letter or deleting a letter results in gibberish sentences that we saw here. Now, if we take a look at actual DNA, we can do the in part of indel, which are what we call insertion mutations, where if we insert an A base here in between the next codon, which would be GCA, the insertion of that base pushes every other base down this way because the reading frame is always in relation to the first start codon that we have. So when we add a base here, in this case the A base, the reading frame is pushed forward by one base. Or rather, the reading frame doesn't change, but the bases that fall within our reading frame are pushed forward like this, meaning that all of the codons here will be shifted downward by one base. And we can see here that this can have very radical effects on the amino acid sequence, because out of the amino acids affected by the insertion, not a single one is the same, and presumably this is true farther downstream as well. Similarly, if we were to remove a base, so if we look at the wild type here, if we were to get rid of this G base here, that means that all of the other bases are pulled this way, because again, our reading frame, where the next codon and the next are, is always based on the start codon, meaning that the reading frame appears to be pulled backward to replace the base that was removed, and not surprisingly, these are what we call deletion mutations, hence why we collectively call frameshift mutations indel mutations, because whether we insert or delete a base, it has an overall similar effect. And the consequences that we can see is that it isn't just a single amino acid that is being changed, as would occur in a point mutation, but basically every other amino acid that follows where the frameshift mutation occurs is probably going to be different. And not surprisingly, proteins that feature a frameshift mutation will almost always be non-functional, or at least will not perform the original function that the protein had. And for an extreme example, this particular protein, which is an enzyme called tyrosinase, referring to an enzyme involved in tyrosine biosynthesis, in its wild type form, it looks like this. But with a single frameshift mutation, we can see that it has radically altered the structure, and likely this enzyme will not be able to function once again. This concludes our series on the types of mutations and the consequences that mutations can have on protein function. To test your knowledge, you can use the codon translation table here and try a few of the practice problems here to test your knowledge of understanding how both point mutations and frameshift mutations can work.